All right, so let's go back to that. So we'll have four instead of five. We will have four topics, so one less. You will see that there are some supplementary material. I highly suggest you to go over those materials. It is directly not, you will not be asked questions from that. But that supplementary material that you have under this module is required because, as I said in the very first class, that virology is an integrative science. You need to remember some of the chemistry, biochemistry, microbiology, initial biology, part of the cell biology. You do not have to take cell biology, but the initial cell biology that you have learned in principles of biology actually helps. So many of these things sometimes we forget, and you need to brush up, and that adds the extra time on top of this material, and that's where the rigor is. And those supplementary materials will actually help. So do your part, you should be fine. All right, let's start with that. Some RNA history, very self-explanatory. We already talked about crystallization of the TMV, 1936. And you can see that 1960s, the studies on viral RNA replication actually began. And that was it. And then there is some fun facts was that. So I am old, you can just imagine that. And then in 1995, as a graduate student, I worked with a double-stranded RNA virus of the Virology family. It's a CPV is a cytoplasmic polyhydrosis virus that I worked as a graduate student. And then the rest of the career I made with the Rhabdoviridis family. It's the vesicular stomatitis virus. And we'll see that we discovered some of these host factors and two entities of the RNA polymerase that we will discuss in the material. Now, if you have downloaded the slide like yesterday before 8 p.m., then there were 3 slides are drastically different. I just put the coronavirus in this material earlier, and then I took it out and just replaced it with the alpha virus, which was already the original part, because I thought the coronavirus I will just use as a special topic, not in the material, because it is going to be too complicated if I just put it in there. All right, so after that, let's go into the RNA synthesis strategy. So here you can see that I put this alpha virus right here and the coronavirus as a special topic. Earlier, this alpha virus was not there. So if you don't have this version of the slide, just make sure that you download that. So here we are going to look at the plus RNA genome, how they are going to make the mRNAs. Then we are going to look at the plus minus RNA genome, double-stranded RNA genome like a reovirus, how they are going to make the mRNA and genomic RNA. RNA replication means we are looking at both, making of the mRNA, making of the genome RNA, both. So, which is also called the transcription and the replication in older terms. And then we're going to look at the minus sense RNA, an example with the influenza virus and the vesicular stomatitis virus. These are the 2 prototypes. So what you are looking at, coronavirus is not a prototype, but we're going to use as a special topic, whereas poliovirus, alpha virus are the prototypes. Alpha virus is, to some extent, very similar, a little bit less complicated, but very similar to the coronavirus, and you will understand that. So these are the main things. So when we're talking about the replication or the synthesis of RNA or the transcription of the RNA in the virus, it was just kind of like a controversy that how these RNA material is actually replicated in the cell, because that was not very clear. So this was David Baltimore, and he was a graduate student at Rockefeller Institute with Richard Franklin. He did, this is the original paper from 1962. So what they did, that they actually have a cell, the cell culture was already discovered 10 years back in 1950s, and they added these ATP, UTP, GTPs, and all the nucleotides. One of them is labeled, radioactively labeled. And they treated the cells with actinomycin D. Actinomycin D is the inhibitor of cellular DNA-dependent RNA polymerases, which makes the mRNAs, which make all the different RNAs. Again, remember that actinomycin D is the inhibitor of the cellular DNA-dependent RNA polymerase. So that, see that in 1960s, that much knowledge of mRNA was already known, and we are getting scared in 2020. So is there any reason for that? So think about that. It was already known, the details of the cellular DNA-dependent RNA polymerases. So now, after that, they infected the cell initially they did the study with a Mengo virus, and then later they did with a polio virus. And it doesn't matter which virus it is, but those are the RNA viruses, both of them in the same family. And they found that if you can see that the red is the single-step growth curve. So you have the initial eclipse period, and then you have the burst. And they measured the amount of RNA in the cell. They extracted the RNA at different time points and looked for radioactivity on the gel. And they found that this is the activity. So the activity of the RNA incorporated with the radioactivity drastically correspond to the virus which actually comes out. And of course, this will drop down because the virus is killing the cell and they are just out of the cell. And so this basically the discovery, seminal discovery, pointed out that even with the actinomycin D, this RNA is being synthesized, so this must be of a different origin. This must be viral origin. And that was the main thing, that the viral RNA-dependent RNA polymerase now that is discovered, the first time they named that RNA polymerase or RNA-dependent RNA polymerase, is not inhibited by the actinomycin D and of viral origin. And that was the start, and then discoveries after discoveries happened. So the polymerase discovered although in the minus strand RNA viruses, and that is the interesting part, that the minus strand RNA viruses actually package the RDRP or the RNA polymerase inside the particle. And we'll explain that why it is. The reason, think about that, the plus strand RNA viruses, when they come into the cell, many of them part of them can act as an mRNA. After the mRNA is translated into the proteins, they can make the RNA-dependent RNA polymerase in the cell, which can do further transcription or the replication. Minus strand RNA cannot do it. It cannot be read by the ribosome. So they bring the RNA polymerase, which is shown just at the very bottom, just right here, and that's the RNA-dependent RNA polymerase with the associated protein. And they do convert these to the plus strand mRNAs so that they can be translated into proteins and also make the genome RNAs. So plus RNA viruses do not have the RDRP in their particles, whereas the minus RNA viruses always carry them. And they do have a conserved motif. These are independent RNA polymerase called a GDD. So you are going to see that you have a glycine aspartic acid motif, GDD motif. That is, all polynucleotide polymerases actually have that. We have a separate slide, we'll see that. And many different studies have been done, many structures solved. So here we are looking at that all RNA viruses do not have RDRP in the particles. Say minus strand RNA genome, RDRP, RNA-coated protein, vesicular stomatitis virus, influenza virus. See that right here, the vesicular stomatitis virus have an unimolecular genome, which is a single genome, one end to the other end, 11,062 base pair, so fixed length. And you have the RNA-dependent RNA polymerase. Whereas the influenza virus have it is not an unimolecular genome, but it is a segmented genome. So you have segments of this, and all these segments are different. So just think about that. Also, don't quote me on that because that will be wrong. But in a sense, you can see that the vesicular stomatitis virus or anyone which does have an unimolecular genome, you can think about it has a 1 chromosome. Whereas here you have 8 segments, think about that you have 8 chromosomes. But again, do not compare chromosomes with that. This is just kind of a very crude analogy with that. So that's your 2 different types of negative strand RNA virus we're going to talk about, RDRP packaged into the particle. Plus strand RNA virus, poliovirus, there is no RDRP and the RNA is naked. The RNA is naked, whereas the RNAs are always coated with the protein and forms nucleocapsid. So why it is so? Think about that. that the minus RNA, as soon as it gets into the cell will sense that this is a foreign RNA. There is no minus RNA in the cell. So it can get degraded. It will just trigger the PKR, protein kinase R, activities, many of the interferon-related defense mechanism and RNase degradation will happen. So that's why they are coded. Whereas the plus sense, as you see, that they are like mRNAs, so that's why they are not coded usually. But there are exceptions. Coronavirus is also a plus strand RNA genome that is very, very large, and that is coded with a nucleocapsid. Whereas the retrovirus is also a plus RNA, but we are not going to discuss retrovirus here because that will be your RNA-directed DNA synthesis. The retrovirus is a separate chapter at the very end. But they also have a plus strand RNA, which is coded. And then you have a double-stranded RNA genome just in a reovirus. They also have the naked RNA. Double-stranded RNA is also not found inside the particle. So how come that they do have the double-stranded genome? Anyone remembers from that? Those double-stranded genome never comes out of the viral core particle. The RNA polymerase is inside the core, and only the mRNAs actually come out of the virus particle. Then what happens, we are going to see that the mRNA and the RNA-dependent RNA polymerase is packaged into the core particle, and the minus RNA is synthesized inside the core particle, and it gets double-stranded. So again, we are going to talk about that in a second. So here, these are the hallmarks of the minus strand RNA viruses. You can see right here, this is the VSV nRNA. So this blue protein is the N protein, or the nucleocapsid protein, and the yellow is the RNA in the groove. You can see that in our N protein is a bilobe structure, and it's protecting the RNA. But it allows the access to the RNA polymerase to replicate it. On the other hand, on this side, you can see that here is just one segment of the influenza virus. Here, the nucleocapsid protein is known as the NP instead of N. Here is nucleocapsid N, NP. So usually these are the names, short names of the proteins. And you can see that how they are wrapped around. And if you look at the coloring of that, the yellow part is always on top of this blue part. The blue part are the positively charged amino acid. And as the phosphates are negatively charged, nucleic acids are called acids because they are all negatively charged due to the phosphates and the phosphate backbones, and they actually wrap around those areas. So here you can see that how it is. And the PA, PB1, PB2, these are the three subunits of the RNA polymerase that we are going to see. So here you are looking at a bigger picture of that. Each one of them is a segment, and at the end you see that there is tiny 3 little dots. Those are your PA, PB1, PB2. So you have all of the segments are associated with the RNA polymerases. So that's how it looks like. All right, so after that, we are looking at that in many of these replication mechanisms, we are going to see a structure which we call as a pseudoknot, and going to see that how this is formed. So you already know that if they have complementary base pair, they can form extensive secondary structure. You have already known that. So let's see that in this structure, a pseudoknot, how it is formed. See that right here, you have some of the bases which are complementary to the bases right here. So this loop falls back onto this and form this structure. And then how the pseudoknot formed, just think about that. You have this S1, L1 loop. You flip it on top of L2 and S2. You just flip it over. And if you just flip it over, just think about this way, then you have a kind of a pseudoknot structure. And the structure of that this is a molecular model which is structurally solved by X-ray crystallography. So you can easily see that that's how a pseudoknot is, and we're going to see that the pseudoknot is a very important structure in the replication transcription in many of the RNA viruses. Particularly, we are going to see the pseudoknot function in the SARS-CoV-2 viruses in the special topic. All right, so rules. Definitely remember that the genome must be copied end to end without no loss of nucleotide sequence, and the viral mRNA produced can be efficiently translated by cellular protein synthesis machinery. That we are seeing right from the very first slide, or the last slide of that with the ribosome picture. We are getting these information all the time. So these are the RNA synthesis rules. So how these things are happening here. So the RNA synthesis initiates and terminates at specific sites on the template, and it may initiate de novo, means just by itself, and the cellular DDRP means DNA dependent RNA polymerase. These are the RNA polymerases. There are many of them. RNA polymerase 1, 2, 3. They make the ribosomal RNA, they make the messenger RNA, and they make the tRNAs. These are your altogether DNA-dependent RNA polymerases. They can just start the synthesis de novo, or they may require a primer. So here we are looking at that you have the template, 3' to 5', and the RNA polymerase is making a 5' to 3', and this is the de novo synthesis. It starts right from the beginning, the synthesis, and you have a hydroxyl end to where you add the bases, basically. This is the 5' phosphate for the next coming nucleotide, which could be A, U, C, G, any one of them. Here you are seeing that a primer is actually formed, and after that primer, the RNA polymerase can take it over, and the process is basically the same. So de novo and require a primer. And sometimes other cell proteins or the host factors are also required sometimes. So that's very important. So the functioning, some of the host factors means that these are cellular proteins that are required for the function of the RNA polymerases in some of the cases. And it is synthesized in a 5' to 3' direction, and the incorporation of NTPs. So definitely underline that word NTP, and we're going to talk about that because it's nucleotide triphosphates. In the quiz question, sometimes it may trick you. I put DNTP. DNTP means deoxynucleotide triphosphate, which is incorporated into DNA, not RNA. This will be definitely your quiz question or exam question. So look at that. For RNA, it is always NTP, never DNTP. For DNA, it is DNTP. So definitely remember that. And some non-templated synthesis also can happen. So we're going to see those. And what we mean by the non-templated synthesis is at the very end, we're going to see some special mechanism of cap snatching and those kind of stuff. We're going to see that. So this is basically a detail of the de novo initiation. So what it does, the RNA polymerase just put the nucleotide 1, the nucleotide 2, and just goes on. So but that's basically what it is. It just doesn't need anything. Then it also need, on the other hand, it is a primer dependent that could be different. It could be a protein primer. So what happens that you have a terminal protein, and the first nucleotide is attached to that protein and put onto the template. On the other hand, it can also synthesize a cap, and we are going to talk about the cap in the next chapter. This is basically, you have a guanosine and adenine structure in there, and you can just go to that wiki link right here and take a look at it. It's not needed here, but cap is a special structure which is read by the ribosome to think that this is an mRNA. So that's the function of the cap and has a special structure. That can also act as a primer attached to that, and you just incorporate NTP. So capped primer or terminal protein, these are the two mechanisms of primer-dependent initiation. All right, after that, we are looking at the sequence relationship of the nucleotide polymerases, and you just can be overwhelmed. It's like what I'm going to just learn from here. So what you're just going to see that the glycine aspartate, so GDD, this is a very conserved motif in the double-stranded RNA viral polymerases. Whereas in the reverse transcriptase, which we are not going to discuss in here, you don't have the glycine, but you have two aspartic acids. So we are going to see that in the last chapter of this module. On the other hand, in the non-segmented minor-strand viral polymerases, you have the glycine, aspartate. These two are conserved, but it is replaced by an asparagine at the third position. So, but basically what you see that these amino acids are conserved in the active site. And where is the active site? We're going to see. Usually what they have, that these are the different RNA-dependent RNA polymerase. This is RNA-dependent DNA polymerase. So remember that. So RNA-dependent DNA polymerase is your reverse transcriptase. Remember that. DNA-dependent DNA polymerase is the eukaryotic DNA replication machinery. In the prokaryote eukaryotes, DNA-dependent DNA polymerase replicate the DNAs. And the DNA-dependent RNA polymerase is also the prokaryotic eukaryotic RNA polymerases that we are talking about, which makes the mRNA, tRNA, and the ribosomal RNA. So just what are the meaning of each one of them? Remember that. So again, the top one is completely viral origin, RNA-dependent RNA polymerase. RNA-dependent DNA polymerase is a reverse transcriptase, which is mostly viral origin. Now remember that we do have, even the prokaryotes do have these reverse transcriptases in your genome. We do make reverse transcriptases, which are the transposable elements that we are going to just talk about that. We talked about 40% of our genome is with these retroviral sequences. And we do have, the humans or the eukaryotes do have these RNA-dependent DNA polymerases as well, just not the first one. So what we are looking at, this A, B, C, D, E, these are some of the conserved motifs in each of these, what we called together, nucleotide polymerases or polynucleotide polymerases. They are very conserved. And the C in yellow is actually the active site. And the active site actually do contain these motifs, glyasp in RT or the glyasp in those areas. So that's basically the overview that what motif you do have, and usually we ask the GDD or the glycine aspartate that motif. So here we are looking at that how the polymerase catalysis actually happens. It happens by a two-metal mechanism. And before that, we're just looking at a nucleotide. So just a recap, you have the base, which could be AUCG, any one of them. Here you have the ribose sugar, and the base is connected to the first base. And you count the carbons like this. You have 1, 2, 3, 4, 5. Remember that it's a pentose sugar, ribose sugar. And then you have the 3 phosphates attached. You have alpha, beta, and the gamma phosphate. This is a nucleotide triphosphate. Remember that, NTP, nucleotide triphosphate is this. This is the building block. When you incorporate the nucleotide into the growing chain, you lose the beta and the gamma. Only the alpha phosphate goes in as a phosphate backbone into the chain. So it becomes a nucleotide monophosphate. The 2 phosphates which are going out as an inorganic phosphate is recycled and will be used up by the cell to make more nucleotides. All right, so how is the two-metal mechanism happens? So here we are looking at a double-stranded, here is an example of a DNA. You have a 5' to 3' end chain, and this is the other way, 5' to 3', the other complementary strand. And you can see right here, you have A, and then you have, just think about that this is reversed, like it is upside down. Here you have the base, and you have this phosphate right here. And this phosphate is attached with this with the sugar, and this is the next one. So what you are basically looking at, this is basically the incoming. You have this alpha, beta, gamma. These are the incoming nucleotide, and this is going to be attached to this end. So you can easily see that this oxygen makes a nucleophilic attack to this this phosphate, and it's deprotonated, and this is coordinated by 2 magnesium ions. And instead of magnesium, in some cases it could be manganese or calcium, but it has to be a divalent metal ion. It is written in your notes, but definitely important. Write it down. It's a divalent metal ion, and usually magnesium, calcium, or manganese. So Ca2 plus or Mn2 plus. Mg2 plus, magnesium is the most common. And you can see that there are 2 aspartic acids, asp-A from motif A and asp-C from the motif C, which is the active site. Coordinate these 2 metal ions so that these 2 phosphates are kept in a proper position for that reaction to occur. And this is basically the function of these amino acids to coordinate these two metal ions for the reactions to take place. So that's the mechanism of catalysis. So here is a very interesting idea that you can see that this is a molecular structure and solved in the poliovirus RDRP. Here is the D238, and it is, remember that the motif is GDD. This is one of the active site motif, and here you can see that you have, so this is the phosphates, you can see. This is right here is the base, and this is the hydroxyl, 2' hydroxyl, which is coordinated by this D238 motif. Remember that, as, right here. So where is that what we are looking at? See right here, this is the 2' hydroxyl. You have 1, this is 2, this hydroxyl. This hydroxyl is kept in place with D-aspartic acid by this bonding. Now, right here, oh, it was right here. So you can easily see that. So this is the NTP. If you put DNTP, which is a deoxynucleotide triphosphate, which is the building block of DNA, just see how a subtle difference will not incorporate it. In the deoxy, why we call it deoxy? Because this oxygen at the 2' position is gone. And that is called, it's a deoxy. So instead of OH in DNTP, it is just H. And if it is just H, it's not going to bind to this D238, and it's not going to incorporate a DNTP. So this is how, at the molecular level, the specificity of the RNA-dependent RNA polymerase is. So what happened in a DNA-dependent polymerase? People have engineered this. They changed that D238 with a tyrosine, and this RNA-dependent RNA polymerase will incorporate a DNTP. So that experiment has been done. So in the DNA-dependent polymerase, again, I'm repeating that instead of a D238, you have a tyrosine here, and if it's a tyrosine, that can bond with the deoxynucleotide DNTP. So again, as I said, that the artificially made RDRP from the poliovirus, by the mutation, it has been made into a tyrosine, and then it has been shown that it can incorporate the DNTPs instead of NTPs. So this much of the knowledge exists at the molecular level. All right. So here we are looking at the structures, and sometimes it was like what I'm going to know. This just have an overview of that structure. But the first thing is that this coloring actually doesn't very much correspond to this color. To some extent it does, but it is a little bit different. So here it is almost close, but not very close, because you do not have this color, this whitish color is not here. But what you are looking at, that if you're just looking at the RNA polymerase, it is known as a 3D pol, and it has like a kind of a right-handed structure. So you can think about this way, and again, it is what people think, that the right side is the thumb, then the yellow part actually is the active site, which makes the palm, and then on this side, you have the fingers. And usually it's a closed structure, it's not open like this, it is closed at the top. And we will mean that what it is. So here, if we just looking at this structure, and again, remember that these colors are, see, here, the previous one, oops, here it is all blue, and the colors try to correspond to these colors, except the cyan and the white. They are pretty much the same. The blue is all of it, and the red is red, the green is green, yellow is yellow. That's your right here, your purple is purple, and the cyan is white. So this is similar. The space filling model is not. This is not the same color as that. So the active site is in the purple, and these are just the different subunits that you are seeing. So what you are seeing, that this is the top view, and if you have a right hand, it looks like this. So this is through which you are going to see the, let me show you the other ones on the other hand. So this is, on the other hand, is the front and front of this. So this is the structure, and this is the structure which is in between the fingers. And see that what happens here from the top, the RNA template actually goes into from the top, and then the RNA template and the product comes out from the front. And the in between the fingers, think about that, there is a very small opening, and the NTPs diffuse from the cytoplasm through these small opening to the active site to be incorporated into the newly synthesized chain. So it is fed from the top and it comes out. So it makes a U-turn, or almost an L-shaped turn, and comes out. So it's fed from the top, comes out from the front. That's how it basically works. Just an overview of that. In between mechanism for exam or quiz is not required. All right, so let's take a look at the different strategies, which is the main part of this topic. So first, we are starting with the Baltimore class 3 double-stranded RNA virus with the reoviruses. So it's a double-stranded RNA. It must make the mRNA that needed to be translated into the protein, and that will make the virus particle. Also, it needs to make the mRNA, which we said that is packaged into the virion, and then the minus strand is made. And again, just to remind you, remember that the green is the plus strand. The green is the plus strand 5' to 3'. This is the convention. The olive is the minus strand. And for the DNA, you are going to see the dark blue will be the plus strand and the light blue will be the minus strand. And this is from the very second chapter when we talked about the infectious cycle. This is the convention all throughout. Again, repeating, the green is the plus 5', 3' strand. That's what the mRNA is. And the olive is the minus 3', 5' strand. All right. So here, how it happens, you have the double-stranded RNA. RNA-dependent RNA polymerase make from each of the segment. Remember that they are segmented. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we're going to see that you can have maximum 12 segments, not more than that. To tell you why. So each of these segments are individually made into this. So think about that. This is just one of the 10 segments. It can make the mRNA that is translated into the protein. On the other hand, the same way, from the double-stranded RNA, it's also going to make the mRNA. Remember that when the RNA-dependent RNA polymerase is reading the double-stranded RNA, it is reading the 3' to 5', reading to 3' to 5' to make a 5' to 3' mRNA. So it's the same thing. Once they make this one, this is packaged into the virion, and inside the virion, the RNA-dependent RNA polymerase is going to make the complementary minus strand on top of this plus strand, and it becomes the double-stranded RNA. So this is your transcription or the mRNA synthesis. This is your replication of the genome. We are going to see this scheme in everywhere. So here, the overview of the reproductive cycle. And the few steps in entry and exit is not required, but just read it one time just to have an overview of that. So receptor-mediated endocytosis, the reovirus gets into the endosome, and see that the red proteins on the outer core is taken off. And this is what is called an infectious subviral particle. It opens up these hydrophobic cores, and it fuses with the endosomal membrane, and it can penetrate into the cytoplasm. And in this process, these blue proteins of the inner core is also lost. Only the very core of the virus is delivered into the cell. This core remains as a core. It does not disintegrate anymore in the cytoplasm. See what happens. While it produces the core, there are some cap proteins on these turret proteins. There are 12 of those in 12 vertices. Remember that it's icosahedron. You have 5-fold symmetry, and you have 12 of these 5-fold vertices, and each of the vertices you have these proteins, which is called a turret protein. And it has a cap on top of that. And that cap is removed when it is the core particle comes to the cytoplasm, and that activates the RNA-dependent RNA polymerase, which is inside the particle. That now makes the mRNA, and all these tiny structures with a black dot that you are seeing, these are the capped mRNAs, and these are going to make all the proteins that is required. And then what happens that as these are pumping out mRNAs, these mRNAs are also going to get packaged into the newly formed core structure. Right here, a newly formed core structure is formed, and these mRNAs are going to get packaged in there as well. See that with all these black dots, they are in there. And then other proteins are formed on top of that core. The core has the mRNAs only, and other proteins are also forming the full reovirus particle. At that time, these mRNAs which are packaged in here is converted into double-stranded RNA by making the minus RNA. So now the matured virus comes out, and it's a naked virus, doesn't have any membrane around with that. It's a capsid, double capsid. And this is how it acquires the double strand. So you can see that this is a strategy. The cell only sees mRNA all the time. Double-stranded RNA is always inside the particle and never exposed to the cellular environment. And this is an escape strategy for them. So it's all written in very detail. If it is a little bit too much, just read the material and rewatch the recording, and you should be fine. So here is just an overview of that. Again, you do not have to memorize the name of the proteins. Just for your reference, just remember that your L1 segment and the M1 segment produce the two proteins, lambda 3 and the mu 2. These two together make the RNA-dependent RNA polymerase. Lambda 3 is the real RNA polymerase, whereas the mu 2 is the guanylyl transferase, or this makes the mRNA caps. So that we talked about all the time. These caps are made by this guanylyl transferase. These two together is the hollow enzyme. So remember that part of the enzyme is called the apoenzyme, and when the subunits all together comes in as a one functional unit, complete functional unit, we call this a hollow enzyme. So your hollow enzyme of the RNA-dependent RNA polymerase is made up of with the lambda 3 and the mu 2. All right, so next what we are going to see. So here, if you're just looking at that sometimes you have a different start of translation that can make multiple proteins. That's one of the important things that you can see. One thing is that, see here, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. A reovirus can maximally have 12 copies of RNA-dependent RNA polymerase which are placed under these 12 vertices. And they can only have 12 segments of double-stranded RNA. They cannot have more than that. And that could be a trick question. If I just ask that somebody reported that there are 14 RNA segments or double-stranded RNA segments in a reovirus, are you going to believe it? Probably not. Because the maximum number that can happen is that each one of them is under one of those 5-fold vertices. And in an icosahedron, you have 12 vertices. That number is constant. You cannot have more than that. That is the architecture of this virus as well. So that's a little bit detail of the protein, that how it happens. That shows the attachment protein of this sigma 1, and that is the turret protein. And what is a turret? So this is a turret. So that's kind of just to give you an idea that what we are talking about. And you can see that the turret is right here, is capped by this sigma 1. And this is the reovirus particle. This is the complete particle. When it becomes ISVP in the endosome, see that the sigma 3 protein goes off. And then you still have this blue protein, which is the mu 1. Once it penetrates into the cell, that mu 1 also goes off, and it also takes off this protein, which is the sigma 1, which is the cap. At this time, as soon as this cap from the turret protein is gone, it activates the RNA polymerase inside, and it pumps out so these are double-stranded from here. It pumps out the mRNA into the cell. So that's basically the mechanism for RNA viruses. And here, if you just want to play around with these, you can just look at these links, that how the molecular structure, every protein of these are completely solved structure should come up. Okay, all right here. So here you can see that you will be able to do a lot of things with these. So all these purple structures are the turrets, and each of these vertices as well. And you should be able to rotate, and it's very slow somehow. So whether it is a computer or the internet, I have no idea. But you should be able to rotate that and look at each of these alpha helices and the beta strand and the side views of those in here. So yeah, so you can just do a lot of fun things that you can easily do by yourself. So that's basically, you can just give the molecular structures. This is a molecular model from the cryo-EM. All right. So the next one is the Baltimore class 4, and we're going to talk about the poliovirus, and here things can get a little bit intense. So the poliovirus is a plus-strand RNA viruses, and the flaviviridae and the picornaviruses are this family, like dengue viruses, yellow fever viruses. These are all plus-strand RNA viruses. And the genome is mRNA. And what happens that for the replication purpose, it has to make a full-length anti-genome. Again, remember that strand full-length complement. This we call as an anti-genome, which is the minus sense. From that template, it gets back the plus sense. And in these, the full length of the genome is actually translated. The full length is an mRNA. So let's take a look at the infectious cycle. What happens if poliovirus delivers the genome? There is a protein attached to its 5' end, and there is a specific mechanism by which that protein is actually removed by cellular proteins. And there is no cap. It's a cap-independent translation, and we're going to see that. And it is going to translate it into this large polyprotein, which is cleaved into many different proteins, which will form the RNA-dependent RNA polymerase as well, and some of them will form the structural proteins. And the RNA replication actually happens on membrane vesicles, and this is just like a crude picture of that. But what happens that, remember that when you have to make these RNAs, you have to make that minus sense intermediate. And you don't want the cell to sense this. So that's why initially they get attached to the membrane. But in reality, what happens is, suppose this is the primary membrane on which you have the plus strand. And when you synthesize the minus strand, you have the secondary and the tertiary membrane. It's shown to some extent, but they completely enclose this structure, so the cell cannot sense that what is going on, or the minus sense, or the temporary double strand inside the vesicles. They cannot see that. So that's basically what it is. And then these plus strand RNAs are packaged into the virion, and the virion comes out. So all the detail is in there step by step in your notes. So here is, again, do not have to memorize the whole thing, but just have an overview of the idea. So here is the full-length genome. You have the VPG protein. This is covalently attached to the 5' end of the genome. Then you have an untranslated region, UTR, untranslated region, and then you have the open reading frame. Then you have a 3' untranslated region as well, followed by these poly sequences. So this, from here to here, this makes a polyprotein, and you can see that you have P1, P2, P3, and these are the proteases. 2A pro, when you see that the protease, this is a 3C pro, these are the proteases, and they cleave this polyprotein into many different proteins. And this is also known as autoproteolysis. The polyprotein cleaves itself, and this is an autoproteolysis function, and you're going to see that word, so definitely underline that word or write that word. It's called autoproteolysis. So you can make this many proteins. Some of them are the viral capsid protein, which is the VP1, 2, 3, 4. These are the ones, whereas most of the others, some of these are the non-structural protein. And right here, the 3D pol is the RNA-dependent RNA polymerases. And now you can just see that why it is called a 3D pol. It's the P3, P1, P2, P3, and this from the P3. This first segment is called the 3CD pro, and this is a protease and the 3D pol together. 3C pro is the protease, and the 3D pol is separated out, and that's how the name comes in. It has nothing to do with any 3 dimension, and it is just from how the structural proteins are associated. And here is the VPG. All right. So after that, what we are looking at, so right here is the VPG, and you can see that this is a phosphate, and then this is the 5' end of the genome RNA. But you can easily see that the VPG is covalently attached by bonding, covalently attached by a phosphodiester bond with a 5' end. But what happens that this covalent bond is cleaved by a cellular protease. Remember that this is cellular, not viral. So this is removed after the plus RNA goes into the cell. And there is a special structure that is formed by a secondary structure, and that is recognized by the ribosome, and that protein, that mRNA can be translated without a cap. This is known as an IRES, or an internal ribosomal entry site. It's around the 749 nucleotide, and we're going to see that once again as well. So again, remember that there is a protein attached to the 5' end, which is cleaved off, and there is an RNA secondary structure which forms which makes the ribosome read the sequence from an internal site, which is known as an internal ribosomal entry site, or IRES. This is a hallmark of poliovirus genome, and without a cap, 5' cap, it can be recognized by the ribosome. These amazing things can evolve with these viruses. So after that, we are looking at this structure of the RNA, and you can easily see that the VPG is still attached and it is taken off, and there are lots of these cloverleaf structures, and this forms these IRES. Also, there is some elements which are called the cis-acting replication element, or CRE. These can move onto the over the structure, because there are lots of areas where you have these complementary sequences, and this CRE element can move over the RNA. And then at this end of there you have a pseudoknot. So what happens, it's very interesting that the cellular polylaminated RNAs are actually not copied. It is only by the RNA-dependent RNA polymerase, only the specific RNAs, which are the plus strand RNAs, are copied into a minus strand. So the RNA-dependent RNA polymerase can distinguish the cellular RNAs, mRNAs, from the viral mRNA or the viral genome to make its own genome. So they are very, very specific. So this is how we are going to see that the minus strand is going to be made by the RNA-dependent RNA polymerase. It's a complicated process, and I'm just going to ask or just have an overview of the idea. So we are just going to look at just this picture a little while. So because we saw this picture, so just think about that. You have seen this picture. In this slide, you are seeing the same thing. So you are seeing the dark blue, so that is your plus strand genome RNA attached with VPG. So what it does, that you are going to synthesize the minus strand from this end. Remember that. This is the plus strand. You are going to start synthesizing from the 3' end, which is where the pseudoknot is, because you are starting at the 3' end. That means the synthesized strand is going in the 5' to 3' direction. It goes the opposite direction. So it ends at the end. So we are going to see that how this starting actually happens. So here we are going to see there is a protein primer dependent synthesis. Again, going back a lot of slides. Remember that we talked about this, that there are some protein primer dependent synthesis. This is what we are going to see in here. So let's take a look what we are seeing. That's protein primer. So how this primer is produced? So on a Cre element, and we're going to see that Cre element, there are two of these 3CD molecules. Remember that it is a 3C and the D, 3D pol is attached together, 3CD pro. Two of them actually attach by complementation binding onto a Cre element, and they melt the part of this stem. Then what happens? A 3D, which is the 3D pol, this is the RNA-dependent RNA polymerase, binds onto this loop structure, and it gets the VPG attached to it, and it attaches covalently. The top one is the RNA-dependent RNA polymerase. Remember that. 3CD pro is not the RNA-dependent RNA polymerase. So this is the whole protein, 3CD pro. This is melting the loop, and 3D pol is the RNA-dependent RNA polymerase, which is that gray part that you are seeing. This is the 3C, this is the 3D. So that's only the 3D. So this is attaching two uridines with the protein. So you have the VPG protein, phosphodiester bond, uridine, phosphodiester bond, uridine. This is the primer. After forming this primer, this whole thing, the RNA polymerase and the primer goes all the way back into the structure of the 3' end. And we're just going to see that, how this whole thing happens. So here you are looking at a membrane vesicle. We said that this happens on a surface of the vesicle. So here you are seeing the membrane vesicle, and here is a protein 3AB, which does have a hydrophobic sequence, and this is the VPG protein attached with it right here. And it anchors with the hydrophobic sequence onto the membrane. Then what happens? Here, another protein, which is the poly-C binding protein, binds to this VPG part. This is VPG, this is a poly-C binding protein. So where is the poly-C? The poly-C is on this cloverleaf structure, the C. These are the these are the C, like the cytidines basically. So poly-C binds in here and brings the genome RNA close to this structure. See that this is the genome RNA with the attached VPG as well. Using these cloverleaf structure, the 3CD pro attaches itself to this, and this will make the primer in that process, and that's what you are seeing. So this is just the basic idea. The 3CD pro is attaching, and also along with that, it will bind to another protein, which is a poly-A binding protein, which binds to the 3' end of the RNA. So so it's getting pretty intense in there. So you have see the 3AB attached to VPG, PCBP attached to the C of the RNA. Here you have the 3CD pro attached to PABP and that attached to the poly-A. After this VPG PUU that we talked about is formed right here, that is transferred and attached to U's. Remember that instead of AT in a DNA, you will have AU. So the last two U's, you have these two attached in there. This is the primer for the RNA synthesis, and it will go on all the way up to the end. And this is basically the structure. So here, a take-home message that what you are seeing, that this is basically circularization of the genome. So the take-home message that for the poliovirus genome replication, you absolutely need this circularization of the genome by these protein RNA interactions, and that is the main theme from that. Usually I will not ask in between protein, in between lines, but that the main thing is that just remember that you need a circularization of the genome to start the synthesis. So how much time do we have? Okay, all right. So the next what we are going to go into that this is another example of plus-strand RNA viruses, which is an alpha virus, and it's a Ptogaviridae family. And here you are seeing a little bit different. So here you have the the RNA, which is the genome. And the difference is that not the full length of the RNA is copied. You have seen that in the coronavirus that we just talked in the first module. The full length of the genome is not copied. But what happens, that we're going to see that in a second. So on the other hand, the full length of the genome is copied into a full length of the antigenome. And from the antigenome, you have these subgenomic mRNAs are synthesized. These are smaller than the genome size. That's why they are called the subgenomic mRNAs. And also, this mRNA acts as the template for the full-length genome. So you may ask that why this is actually happening. This terminate at certain sequences in here because of these slippery sequences, because the RNA-dependent RNA polymerase falls off at certain size, which basically makes these subgenomic mRNAs. So here in the whole sequence, that what happens, you have this mRNA, genome RNA comes in, which makes a polyprotein, which is cleaved into the RNA-dependent RNA polymerase. There are several entities with that. And the genome is associated with the, acts as a template to make the antigenome on the vesicle. On top of that, it is also made into the full-length genome RNA or subgenomic mRNA, which is translated again into the structural protein. Here you have the non-structural protein formed in the first instant, and in the second set, you have the structural protein formed. Remember that this is the difference between the poliovirus. All the proteins were formed from the whole length of the genome RNA at one instance. Here it is in two steps, and the structural proteins are formed from the subgenomic mRNAs. And the capsid and the other proteins are associated with the ER. These are the membrane-bound proteins, and they assemble and the virus actually exits. But this central part is more important, that two types. And we are going to see that basically this is the overview of that. You can see that this is the genome. The 5' end is the non-structural ORF. And these are the proteins formed. These are the non-structural protein, including the RNA polymerases. The 3' end has a structural ORF, and this forms the subgenomic mRNAs to form the structural protein, and also the minor strand helps in the genome or full-length genome RNA synthesis. And this is just a duplication of the previous slide, that what you are basically seeing. So this is the overview of that, and we will stop here. And you can easily see that it is just a lot of material. So just go slow, read the material underneath. It is very detailed. You will see the notes are in very detail so that it makes sense. Remember that you may like, how I'm going to read these notes. Remember that you don't have a textbook. That's why I just almost gave you all the material. So that's the reason. Textbook means like, if you have a textbook, you will have the whole material, but many of you don't have. And that should not cause any problem. You have the material, but that's what it is. So if you have any questions, let me know, and that's it pretty much. So have a good one. We will see you Thursday.